Welcome to Decoding Healthcare Research. Join us as we delve into the behind-the-scenes world of groundbreaking research and the dynamic healthcare industry, interviewing top paper authors, engaging experts on industry-related topics, and exploring medical subjects that affect our daily lives.
Welcome to Decoding Healthcare Research. I'm your host, Efrain Riveros, Dr. E.F. Rain. Continuing talking about exercise and the metabolic effect of this important therapeutic, if you will, maneuver, we will discuss a little bit about the role of exercise on fat metabolism, specifically brown fat. So we have the honor of having two researchers here who have done a lot of research about the topic. We have Dr. Henver Brunetta. He's a researcher of the University of Campinas in Brazil. And we have Dr. Logan Townsend, also a researcher at McMaster University in Canada. Welcome, Dr. Brunetta and Dr. Townsend.
Thank you.
Thank you very much.
Thank you.
It's a pleasure to be here.
Thank you. Thank you so much for accepting the invitation. So first of all, I want to explain the audience a little bit about what is the brown adipose tissue. We are very familiar with the importance of fat when it comes to exercise and obesity and metabolic syndrome, but less attention has been paid to brown adipose tissue, at least for the general audience. So I will probably start with Dr. Brunetta. Can you tell us a little bit about what is brown adipose tissue and why is it important?
Yes, sure. So brown adipose tissue is a specialized adipose tissue depot that what we found on that adipose tissue is one of the major characteristics expend energy in form of heat. So one of the hallmarks of this tissue is the presence of a protein called UCP1, and this protein can allows the movement of protons into the mitochondria, and then as a result, you expend energy, you increase oxygen consumption, and generates heat. This was found initially in animals who need to generate heat in order to survive, also in newborns, and then over the years, it was also found in adults, in humans. Another characteristic is a huge presence of mitochondria that we don't have too much in white adipose tissue, and also the distribution of the lipids. So instead of white adipose tissue, we have one big lipid droplet. In brown adipose tissue, we have several very, very small lipid droplets. So that's the main characteristic of brown adipose tissue and the role in thermogenesis to keep the body temperature in small mammals mainly, but then over the years we discovered this also is present in humans.
And in terms of metabolic pathways, what differentiates white adipose tissue from brown adipose tissue? And are they, can the white adipose tissue convert into brown tissue, or whatever amount you have of brown adipose tissue is what you have for the rest of your life?
No, that can change. So we know that different lifestyles can change the amount of brown adipose tissue. Most of this research was done in mice, so pretty much everything we know is from mice, but they can, in some depots of white adipose tissue, you can have markers of brown adipose tissue. That's what we call browning or beiging, if that's coming from white adipose tissue. And on the other hand, generally people with obesity or sedentary people, there is an association that those people have also low brown adipose tissue. So it's not that once you have you can change the amount of the activity of this tissue. Apparently, depending on the lifestyle or the conditions, that can be changed.
I see, and that actually acquires importance when it comes to exercise. But I assume that whatever happens in the adipose tissue is not necessarily triggered at that same level. Probably when you do exercise, there are multiple connections between important organs, the muscle probably being the most important. And this question, I'm going to ask Dr. Townsend, uh, what happens in the muscle that triggers these metabolic changes in the adipose tissue, specifically in the brown?
So that's a really good question. There's probably close to a dozen secreted factors from skeletal muscle that have been identified that seem to be able to do something to brown adipose tissue. How much of a contribution skeletal muscle makes to brown adipose tissue, I think is very complicated, because lots of tissues secrete factors that have been shown to control brown adipose tissue metabolism. And there's probably direct connections from the brain to brown adipose tissue that are really important as well. So, yeah, go ahead.
Okay, so in the paper, you guys start analyzing one. I think it's one of the landmark papers published by Dr. Kim and Associates in 2022, where he develops, I think it's an animal model, right? And he postulates a mechanism involving kinase responsive to AMP and IL-6. And you guys have some comments about this cycle. So can you elaborate a little bit more about what is exactly what happens when interleukin-6 is produced, and what did Dr. Kim say in his paper?
Yeah, so the mechanism that they identified in the original paper was that when you exercise, you get an increase in the activity of this protein AMPK, which we've known for a long time. So when you exercise, you generate a ton of energy, and AMPK is kind of the energy sensor of the cell, they'll say. So, and then what they went on to show was that activating AMPK can promote the release of interleukin-6 from skeletal muscle, which then circulates in our bloodstream, and then causes the activation of brown adipose tissue. So that was the idea that they proposed.
So the order would be interleukin-6 produced in the muscle, and then AMPK activated in the adipose tissue. Is that correct?
No, it's the other way around.
Okay.
So AMPK is activated in skeletal muscle during exercise, and according to this paper, that could be the mechanism or the signal to drive the skeletal muscle to release this cytokine, and then the cytokine, once in the blood, could reach brown adipose tissue to induce the adaptation from exercise.
And when you talk about adaptation, what is exactly happening once IL-6 gets to the adipose tissue, specifically when we are talking about brown adipose tissue? What happens inside that cell?
So that's a really good question, and that's one of the things that isn't super clear. So it seems that cells that are exposed to IL-6 increase kind of these classic browning markers. So like Enver mentioned, UCP1. But how IL-6 does this really wasn't clear, and we really don't know. And that kind of goes back to the point that there's a number of factors that all seem to have these same effects, but how or why really isn't super clear yet.
Isn't clear yet. And yes, for our audience, UCP1 refers to an uncoupling protein 1, right? That uncouples the oxidative phosphorylation and starts producing heat. Is that right?
Yes. So UCP1, as it was called back in the day thermogenin because of thermogenesis, but it's uncoupled the activity of electron transport chain from the ATP production, and that's why you generate heat, and it increase exponentially the energy expenditure.
And when that happens, is that contributing to weight loss, say when you do exercise?
So that's one of the proposals that of the mechanism of weight loss induced by exercise, but the contribution of brown adipose tissue to whole body energy metabolism is quite small. In rodents, this study was done in mice, so in rodents the contribution is much bigger than in humans, but in both, when you're talking about that skeletal muscle, we're talking around 40-45% of body weight, and you can increase the energy expenditure of skeletal muscle when you're exercising by tenfold, right? So you have the mass and you have the capacity to increase energy expenditure. Whereas on the other side, it has been proposed that exercise inducing in adaptation in brown adipose tissue, and that could help the body weight loss that you can see in exercise, but it's not been proven yet.
I see. And everything we have talked about so far, revolves around IL-6, but there are other myokines, some of them better characterized than others. But in the paper, I read something about the role of irisin. So what is that exactly, and how can we make it fit into the equation?
So irisin is another myokine. So similar to IL-6, the idea is that when muscles contract, you get the release of these secreted factors. So IL-6, irisin, same thing. And then they enter our bloodstream, circulate to other tissues, and then they have effects there. And irisin, again, was one of the earlier ones to kind of show this muscle fat crosstalk idea. So irisin has also been shown to cause increased adipose tissue thermogenesis.
In humans as well?
There's some evidence that it exists in humans, and that it does respond to exercise. You get an increase in circulating levels with exercise, and this can be associated with some markers of thermogenesis. But again, in humans, it's a little more difficult to make conclusive claims.
I see. So IL-6 is the better characterized out of all those. But when you read about the topic, you find that there are exerkines that not only are released by the muscle, but other tissues. So which ones because there are many, which ones are potential candidates to actually participate in the process?
So there's irisin, there's meteorin-like, there's FGF21, there's even glucagon has been some evidence, even other ones like GDF15. There's dozens of exerkines at this point, and there's always more, and they've all have some evidence to suggest that they can cause the browning of adipose tissue. Their overall contribution is really difficult to determine. Personally, I think probably one of the main ones are still our catecholamines, so epinephrine or epinephrine. I think those are probably the major contributor, and then all of these other ones probably play complementary roles or have other functions that contribute.
But based on the research that you guys are doing, do you think that that the browning of the fat is something just biochemically sound, but is not really clinically important, or you think that this is definitely part of the beneficial effect when it comes to exercise and other strategies like cold plunges, fasting? There are many interventions that use the same pathways, but is this something just happening in the lab or in animal models, or you think that is actually happening in real life?
So that's a really good question and really important. And so, let's start from the lab. So you can definitely see these adaptations in mice. There's dozens of papers that if you exercise mice, you get increased adipose tissue thermogenesis. But what's really important is that, or what's becoming clear, is that this might be kind of an artifact of the way that these experiments were done. So these earlier studies with mice, the mice live at the same room temperature that we live at. So room temperature, 21 degrees. But for a mouse, that's actually quite cold. So mice prefer to live, maybe 25 to 30 degrees Celsius. And actually, if you house mice at those same temperatures, then all of a sudden these adaptations don't occur anymore. So then if you exercise train mice, they don't show evidence of browning anymore. And this has been shown multiple times. So it might be this interaction between cold and exercise that led to this happening. And that's what's important, is that because in humans, there's very little evidence that you get a big increase in adipose tissue thermogenesis with exercise. So this kind of reflects now what we're seeing in the mice and in humans. We just needed to do the experiment a little bit better. And so overall, I think that despite the fact that humans don't really get a lot of browning when we exercise, we still certainly have a ton of benefits from exercise, right? So in that regard, I think probably it isn't a huge factor for the benefits of exercise. And I think that Enver probably has so a little more to add to that.
Yeah, sure. And the other point, which is very important to highlight here, is you can have beneficial effects of exercise without losing weight, right? Because when you think about thermogenesis and energy expenditure, the main point is fighting obesity, which is definitely a valid point given the epidemic we live nowadays. But when you go to exercise, you have an effects in cardiovascular system, you have effects in the brain, and so many others, you can name it, that are not that are independent of body weight loss. So that's very important to highlight, because you do have beneficial effects of exercise without being necessary to body weight loss.
Yeah, that's true, and I want to highlight the point that you guys made, and what Dr. Townsend said, that the experimental conditions are critical when it comes to animal models, because just a tiny change in the temperature has huge metabolic and biochemical effects that definitely can change the results. And that's I think the most valid point when you made the critique for the paper of Dr. Kim, is that that particular environmental temperature setting might definitely obscure the results. And again, even if exercise is not, or the benefits of exercise are not completely explained by browning, that doesn't mean that exercise is useless. Quite the opposite, because it just uses different pathways, and that probably takes me to the other paper that is a little bit more specific. And I'm gonna actually read the title of the paper. It's a mini review published in the American Journal of Physiology, Endocrinology, and Metabolism in 2020, mitochondria-associated endoplasmic reticulum membranes in glucose homeostasis and insulin resistance. And I brought this paper to the table because when we talk about obesity, metabolic syndrome, exercise, everything comes down to the mitochondria, and what people talk about is always mitochondria, biogenesis, autophagy, and all these terms. But the endoplasmic reticulum hasn't received the same amount of attention. And the paper that you published talks about actually a shared membrane between the mitochondria and the endoplasmic reticulum, and how this organelle is critical when it comes to insulin resistance. Can you tell us a little bit more about that?
Yeah, sure. So that's a very interesting topic, and that comes back when Dr. Townsend and I used to work together at the University of Guelph in 2018-19. So I am expert in mitochondria, while Dr. Townsend at the time was working with endoplasmic reticulum. It is an expert in that area, and we used to talk a lot about how we address these questions generally as scientists, which makes sense because you have to pick one organelle, you have to pick one hormone, one system, but that doesn't mean those things are independent of each other. And that's why we decided to talk about that and to study the interaction between endoplasmic reticulum and mitochondria. And what we know now is you have a physical interaction. That's the MAM we talk about in the paper. So mitochondria associated membranes. We have structures, proteins that can physically connect endoplasmic reticulum and mitochondria. But it's not just about the physical connection, but also about the functional connection. And what we know now, even though this area is not, we don't have too many papers, so there are several open questions in this area. But when it comes to insulin resistance and obesity, this interaction, physically or functional, is somehow changed in models of obesity and insulin resistance or diabetes, you name it, right? So what is interesting is apparently what you find in obesity in liver is somehow quite the opposite what most people find in skeletal muscle when it comes to the interaction between these two organelles, right? So I believe that, and I don't know what Dr. Townsend has to say about that, but I believe there's still several open questions in this field. But what we know is in some cases, when you target this interaction between mitochondria and endoplasmic reticulum, several components of the cellular function are improved or rescued from these conditions of insulin resistance. And it could have a therapeutic angle in the future, but much more work has to be done as far as to fully understand how those two organelles work together.
Yeah, and that was actually my next question. So the finding is absolutely interesting, and it basically focuses on the internal communication between organelles, being the mitochondria and the endoplasmic reticulum critical for the cellular function. But what should come next in order to make this finding useful in the future and to incorporate it into the understanding of the metabolic pathways during exercise and the pathological conditions such as obesity and metabolic syndrome?
Yeah, so like Dr. Brunetta said, it's what we need to figure out first is what is the relationship between these two organelles and insulin resistance. It's really complicated because it seems to be different in different tissues, and these interactions are also really dynamic. So you can get them moving closer together or farther apart, depending on whatever stress you have, whether it's obesity or exercise. And we really don't know whether we want them close together, far apart, or whatever at this point. So really, we need to just characterize this relationship better and in response to different things. I don't actually know if we know what happens with exercise yet, let alone in different tissues. So there's a ton of work to be done there. And then we still need to figure out what are the most important measurements, because not just distance, but functional interactions. So whether they're close or far apart, but then what's actually occurring at this intersection, and what's the most important role of this interaction. So until we kind of get a better handle on what they're doing, it's kind of hard to say how we should be targeting them.
Yeah, I agree. But it's important that the interaction is starting to be characterized, because it probably is going to give us the key to some mechanisms that are not yet fully understood when it comes to the mitochondria, and the answer may be in the interaction with the endoplasmic reticulum. And I think these two papers have actually helped us understand a little bit better what's happening metabolically speaking in conditions when the equilibrium is lost, like obesity, and also with the intervention of exercise. And I appreciate you guys sharing your knowledge with me today and with our audience, talking about this important topic. So first, I want to thank you for being here today with us and sharing your knowledge.
Oh, that's our pleasure, and thank you for the invitation. And we're here anytime if you want to chat again. It's our pleasure.
Excellent. Actually, we are going to follow up on your research, and as new results emerge, we will definitely contact you guys to update the results and everything that you guys are working on. Thank you for sharing the message, and for our audience, don't forget that in the description of the video you can find the links to the papers and some information about the authors. Thank you for being with us today, and see you next time.
Thank you so much for tuning into this episode. If you enjoyed it, please don't forget to give us a thumbs up and share it with your friends and family. Make sure to subscribe to our channel and hit that notification bell so you never miss an episode. If you have any questions or thoughts about today's topic, we'd love to hear from you. Feel free to leave your comments down below. For more information and references related to today's discussion, you can find them in the video description below. We appreciate your support and look forward to having you back for our next episode.